Serene Kwan from St. Mary's High School, and she's been working in the Johannes Ye's lab under the guidance of Johannes Ye.
Hi, I'm Serene Kwan from St. Mary's High School, and I was mentored by Dr. Ye. And our project was focusing on the expression and purification of wild-type and oncogenic mutant of KROS inhibitors. So what is a ROS? ROS is a protein and it's also a type of a small GTPase. What a GTPase is, it goes through a hydrolysis cycle and it's on when it's at GTP bound form and it's off at GDP bound form. And when it's on, it sends signals inside the cell to stimulate cell growth. And when there's a mutation in the ROS, it could be a cause of cancer. So mutated ROS is found about 30% of tumors. And our project goal is to purify the wild-type KROS and the G12C ROS for developing an inhibitor for the mutations. So how does ROS signaling work? When the signal molecule comes to the receptor, it phosphorylates the tyrosines at the tail of the receptor, and then the adaptor protein binds to the tail, and that recruits the ROS activating protein near the membrane because the ROS is also at the membrane, and then the ROS activating protein makes the inactive ROS to drop the GDP, and it makes the GTP go on instead, and the active ROS sends growth signals in the cell, and after that it has to go back to the GDP form by dropping a phosphate, but there's a problem when that does not occur, and when ROS mutations occur, it doesn't go back to the GDP form, and it keeps sending signals within the cell. So we focus specifically on the G12C mutation. That's a mutation where the ROS hydrolysis occurs very slowly, and it keeps remaining on the GTP form. So therefore, our goal is to purify the wild-type ROS and the mutated KROS G12C to inhibit the ROS mutation. So to purify ROS, we first have to express the gene. So we introduce IPTG into the E. coli genome. We're using the Rosetta cell in the experiment, and then that produces the T7 RNA polymerase. And the T7 RNA polymerase attaches to the T7 promoter of the PET plasmid, and the IPTG goes here, and then we can get our target gene from the plasmid, which is the ROS protein. And we're also using the antibiotic kanamycin because the PET vector has a kanamycin resistant gene, so therefore we could only select the cells that have that resistant gene by using kanamycin, because all the other cells that don't have the gene will die off. And for the same reason, we're using chlorophenicol because the P rare plasmid from the Rosetta cell has 6 rare tRNAs, and that helps the translation process go smoothly because it supplies the tRNAs that aren't in there. And the P rare plasmid has a chlorophenicol resistant gene. So for the same reason, we're only going to keep the cells that have that plasmid. And after we select the plasmids, we grow, and we have 2 plasmids in the Rosetta cell, and then we start at a low concentration, and through culture we get up to here. That's where we introduce IPTG, and the optical density has to be between 0.8 to 1 when it reaches this point. And now that we express the ROS protein, we now have to purify it. So how we're going to purify it is we attach a His tag to the ROS protein, and that histidine, it binds to the nickel. So this nickel over here, and this is the histidine, and we capture the ROS protein through that process, and we wash the other stuff off. But we want to separate the nickel and the ROS protein. So how we're going to do that is we use imidazole. Imidazole has a similar structure to the histidine, is shown here, and when we put imidazole in there, when we elute with it, it competes with the histidine, so it attaches to the nickel instead, so we could separate the nickel and the histidine, so we could separate it over here. And then after that, we ran the gel after elution, and this is our result. The ROS has a size of around 21 kDa, so we see it over here, and in the ends, at E end, we have some impurities. And we did the same for the G12C KRAS. And this, in the end, the last lane is the wild type ROS, and why we have it over there is we ran the gel again after dialysis to see if it was still there. So what is dialysis? Dialysis is the process of getting rid of the imidazole. After elution, we still have the imidazole, but we only want to keep the pure ROS, and that's why we do this. So if you put it in the membrane, this membrane has a size where only the imidazole can exit, but the ROS can't because the ROS is bigger than the imidazole. So through the process of osmosis, the imidazole will leave the membrane and ROS will stay inside. And we have to switch the buffer like 2 to 3 times to keep the buffer clean. And then in the end, we get clear ROS. And after that, we have to purify ROF. So what is ROF? ROF is a protein that's activated directly after the ROS is activated. And how it works is when the ROS, the active ROS, come near the membrane, it attaches to the RBD of the ROF. RBD stands for ROS binding domain. And then that opens up the ROF. It was first in an auto-inhibited complex, but it becomes open. And then the open ROF then signals the MEK, which is the protein that follows right after it, and it keeps sending the row signals like that. So if we could inhibit the ROF, that could also be a way of inhibiting the ROS mutation. So this is our result from purifying the ROF, and this is our result of purifying FG211. FG211 is a negative control because it's a protein that's totally irrelevant to both ROF and ROS, so it shouldn't show any interaction with both proteins. And to proceed, we have to exchange the nucleotide on the ROS. That's because after ROS is purified, the nucleotides are mixed, and you want to differentiate them because some drugs only target the ROS GTP form, and others only target the ROS GDP form. So you want to have a uniform conformation, and you want to differentiate them. And to do that, you first have to remove the magnesium chloride from the ROS because it impedes the nucleotides from being released. And after you get rid of that, you load a lot of amount of your target nucleotide, is either GTP or GDP, but in the drawing I use GTP as example. And then that goes in place of the original one, and it attaches to the ROS, and we have to lock it again by adding magnesium chloride. So after we differentiate them, we exchange the nucleotide, we have to incubate it with ROF. And as I mentioned before, ROF only attaches to the active ROS. So the ROF attaches to the ROS at GTP form, and it doesn't attach to the GDP form. We're gonna do this through the Nickel piece again. We're still in the progress because we didn't get here, but I expect that the ROF will only show when it's attached to the GTP and not at the GDP. And FG211 doesn't show at all because it's a negative control and it doesn't interact with neither the ROS or the ROF. So for our conclusion, we successfully purified both the wild type and the G12C mutant KRAS and the ROF, and our goal is to target both forms, the GTP and the GDP ROS, with the inhibitors. And also, although we're still in the progress, we want to differentiate the ROS in GDP or GTP form with the interaction of the ROF. And both proteins, ROF and ROS, have future potentials of being used as possible drug candidates for inhibiting the ROS mutations. Thank you.